I'll go ahead and get started. Well, thank you, Nicole, for that introduction, and thank you to the committee for inviting me to come down and talk about some of the research we've been doing in Southern California chaparral communities for the last few years. Most of the work I'm talking about today is work I did as part of a dissertation at UC Berkeley, and some work subsequent to that. I got my start in chaparral ecology as an undergraduate student, doing work with Dr. Stephen Davis at Pepperdine University, and studying the impact of mule deer on the ability of chaparral ecosystems on the California Channel Islands to recover from fire. And from that, we learned a lot of really interesting things on the islands that have continued to spark new questions. And they're all under this umbrella of global change, environmental change. And one of the things that really inspired me as an early career researcher about the chaparral is its incredible resilience and persistence in the face of environmental change. This is me when I had a lot more hair as an undergraduate student, working out on the California Channel Islands and looking at the effects of mule deer on post-fire recovery. But from those early experiences, I was incredibly impressed with the chaparral's resilience, its ability to tolerate these incredible droughts that we have here in Southern California, and to recover from high intensity stand replacing crown fires. And those early research experiences also showed me, though, that there are sometimes limits to that resilience. And so a big part of my research program is trying to understand where those limits are and what are those kind of key mechanisms that drive resilient ecosystems into other states. But before I talk any more about the plants of the chaparral, I just want to take a second to acknowledge the people of the chaparral, especially those that have been influential in my research and in my career. My undergraduate mentors, Steve Davis, Brandon Pratt, and Anna Jacobson, who taught me to appreciate the natural history and ecology of these ecosystems, and to see the beauty in them and to see the resilience in them in ways that most Southern Californians don't get to see. My graduate and postdoctoral mentors, David Ackerley and Max Moritz, who do a lot of great work in chaparral ecosystems across the state. And Marty Witter, my mentor for the Climate Change Youth Initiative, George Melendez Wright Fellowship Program in the Santa Monica Mountains. These people were really influential in getting me to appreciate the chaparral, and I hope to pass that along to my students as well. So in preparing for today, I was thinking about some of the documents that Nicole sent us, and this quote jumped out to me as kind of being key to what we're trying to talk about in the next 3 days, focusing on the ecological vulnerability of chaparral landscapes, what managers might do to promote chaparral persistence and resilience in the face of environmental change. And so for me, there's 3 themes within that's not what that's supposed to do. Within that really jump out as being relevant to the kind of research that I do. But vulnerability, understanding what that is, going beyond just this kind of subjective concept of vulnerability, and how do you actually measure that? How does it vary across the landscape, across space, and across time? What are those examples of persistent and resilient chaparral communities, and when does that break down? So these are going to be themes for the talk today. So I'm going to start, because I'm a new professor, I take a little bit of joy in pop quizzes. So does anyone know what this plant is? Ironwood. Ironwood, awesome. And where does ironwood live? On the Channel Islands. Is it only on the Channel Islands?
Today.
Today it is. What does that mean, today?
It used to be everywhere, all over the Southwest.
It used to be other places, right? And now we can only find it on the California Channel Islands. So ironwood is an example of what we call paleo or relictual endemic. It's a species that was once more widespread. Its range has contracted, and it now only persists in certain environments. We know this because we find fossils of ironwood in places that we don't see ironwood trees on the mainland across the western US. And this concept is especially important here in Southern California in thinking about the flora of the California Channel Islands and what makes them sort of unique from the rest of the landscape. But the basic idea that plant species can have this wider distribution, that over time that distribution can be restricted to certain environments, and that the current patterns of distribution are a function of persistence to past environmental changes. So thinking about those themes of the talk today, or of the conference this week, we have examples here in this landscape of persistence. It's a natural part of chaparral landscapes. And these are models that we can study to learn what about these systems make them resilient, allow them to have species that are persistent in the face of environmental change. So the ironwood is one example, but Daniel Axelrod noticed that a large portion of the flora from the California Channel Islands follow this pattern. Climate became drier and more intemperate over southwestern North America during the Miocene and Pliocene. As a result, species allied to the insular endemics occupied a progressively shrinking belt of highly temperate climate that was gradually restricted to the California coastal strip and islands. So again, in this landscape, we have examples that we can draw from about persistence and resilience. So thinking about what makes the island sort of relatively unique to the Southern California landscape, their climate is a little bit different than what we see here on the mainland, especially interior portions of the mainland. We have similar mean annual climates, similar mean annual temperatures and precipitation, but the extremities of the temperature are different. We get higher temperatures in mainland environments. That moderating effect of the ocean maintains lower temperatures, especially crucial during those long protracted dry seasons that we have here in Southern California. In addition, there's an increased presence of fog in island environments, and this all works together to create a relatively cool and foggy climate in the midst of the rest of the Southern California region. Okay, so switching gears a little bit, that's a little bit about the islands and why they might be interesting to studying persistence and resilience. But going back to this idea of vulnerability, how do you measure vulnerability? We're fortunate here in the Chaparral that a lot of research has laid the groundwork for us to understand and be able to quantify vulnerability in very specific ways. And it all relates back to the way that water moves through plants. So if you think about the way water moves through a plant, it's this passive system where the water is evaporating from the leaf surface, and that pulls water in from the soil into the plant roots and up through the plant and out to the atmosphere. This is an incredible process that is capable of moving water short distances in very small plants, but is the same process that moves water from the soil all the way to the top of the tallest redwood tree in the world. So it's an incredibly efficient and effective way of moving water long distances. That said, it has some limitations. This system is operating under tension, and the water molecules form a continuous column all the way through the cells, those specialized cells inside of plants that are specialized for moving water. And those water molecules are joined to each other and and they adhere to the walls of those conduits, and it creates this continuous column. And the suction creates a tension on that water column that in certain conditions can pull air into the system. And when air gets pulled into that system, it changes the function of that cell. It can no longer transport water. This air blockage is something we call cavitation or embolism, and we can measure it. We have developed ways here. A lot of the work to do this has been developed here in chaparral ecosystems. And we can measure it with something called a vulnerability curve. Oh, you can see it there. I can't see it. Okay, this is a vulnerability curve. So what are we looking at here? I always tell my students, if it doesn't have labels on the axis, it's nothing. It's just a couple of lines. So we're going to label the axes here. The x-axis here is showing us tissue dehydration with increasing dehydration as you move from right to left. So 0 means very hydrated tissues. -14 is incredibly dehydrated tissues. And we can also measure the function of the hydraulic system as the tissue dehydrates. And that's what we're depicting here on the y-axis. And so you get increasing loss of that function as you move up that y-axis. So we can generate these for plants that live in chaparral ecosystems, and we can quantify what their vulnerability to drought-induced cavitation is. One way we try to standardize these curves and compare them across species and across environments is to look at particular parts of the curve. In particular, we look at the point at which 50% loss of hydraulic function occurs. And we look at the water potential where that occurs. We call that P50. And then it's also helpful to have an understanding of what the plants are actually experiencing in the field. So we can measure their tissue water status in the field, and we can compare that to these vulnerability curves. And by looking at the difference between the tissue water status in the field and what the plant can experience before it loses half of its ability to conduct water, we can measure something called the hydraulic safety margin. So it's that difference between what the plant can experience and what it is actually experiencing. And these traits are things that we have measured in many different sites across the chaparral, and they help us understand vulnerability and how it's changing across space and across time. Now, for a particular species or a particular population, these things are not static. They can change. You might have a situation where the minimum water potential is actually much more favorable than in other situations, and that widens that hydraulic safety margin. Okay, so it increases the amount of additional dehydration that has to happen before you have dysfunction of the hydraulic system. You can also have a situation where vulnerability shifts. That vulnerability curve changes its shape. This can happen ontogenetically between young and old tissues or across sites or across species. And that can also be important for what that safety margin is, what that vulnerability actually looks like. And so a big part of my research was asking this question. Are island plants, we have these examples of islands being these places where relictual endemics can hang out, is one of the mechanisms that allows for that persistence on islands related to this idea that islands are less vulnerable to something like drought-induced cavitation, particularly in the semi-arid environments here in Southern California. So to study this, we matched up field sites from the California Channel Islands and the mainland, and we tried to keep as many things constant as we could. What the mean annual climate, so similar mean annual temperatures and precipitation, slope, aspect, elevation, soil characteristics. And importantly, chaparral communities. These are both chaparral communities occur, both on the islands and on the mainland with similar species assemblages. And so we wanted to look at these particular traits across a range of different species in these communities to see what the differences are. And here's what we find. So if we look at the minimum water status of island plants compared to mainland plants, island plants have a higher, what we call leaf water potential, so more favorable tissue water status at the end of a dry season compared to close relatives living on the mainland. But when we look at cavitation resistance, so that P50 metric, that shape of the curve, we find no detectable difference or no repeatable pattern of difference between the island and mainland plants. So what that means is the plants are experiencing less seasonal water stress, yet they're just as capable at tolerating water stress as their mainland relatives are. That's really interesting. And as a result, they have wider, on average, safety margins. So if we look across a range of different functional groups and species pairs from island and mainland environments, the island plants have consistently more favorable safety margins. So they have a wider operating range before they experience hydraulic failure. So that's sort of into the nitty-gritty of the physiology of these plants. So then what is that predictive of how these island plants might fare during recent drought events? And thanks to climate change, we have some good opportunities to look at how plants are actually responding to recent droughts. This is showing percent area in drought for the Southern California coastal area, according to the US Drought Monitor. So those different colors there represent different levels of drought: exceptional, extreme, and severe. And basically you can see what many of you in this room probably already know, that we've had several single-year droughts since the year 2000, in 2002, in 2007, and then this more recent multi-year drought from 2012 to 2016, 17, depending on where you are. So this provides us an opportunity to take what we know about the physiology of island plants and ask this additional question: did the island plants fare better during these drought events than mainland plants did? And to do that, we are using some remote sensing tools to look at drought stress from space using satellite imagery and looking at the reflectance patterns of plants. And healthy plants and unhealthy plants have different patterns of reflectance that we can observe from satellite data. And we can use that to generate these vegetation indices. A common one is the normalized difference vegetation index. You may be familiar with it. We are using something that's very closely related called the enhanced vegetation index. And so I want to point your attention to this time series of enhanced vegetation index for island and mainland environments. So we looked at polygons covering most of the Channel Islands and found comparable sites on the mainland. And basically what we see is that during these crucial drought events, the acute drought of 2007 and this more protracted drought in more recent years, the island plants maintain more favorable reflectance patterns, or show that they're healthier during these drought events than comparable sites on the mainland. So this provides an additional test of that idea that island plants are maybe buffered from some of the regional drought exposure that this region experiences. So we've been talking mainly about island environments, but the take-home message here is that drought vulnerability depends on where you are in the landscape. The islands are a little bit less vulnerable than the mainlands are, but that's not the only place where that might be the case. This is a recent paper from Blair McLaughlin and colleagues out of Global Change Biology, arguing that these kinds of environments, these what we call drought refugia, or places where drought experience is going to be a little bit more moderated relative to the rest of the landscape, are all around us, and they're going to be increasingly important in the 21st century as a result of climate change. So that blue box there is showing you some of the processes involved with things going on the island. But there are many other environments within the landscapes where we live where this kind of buffering might occur. In addition, there is starting to be some movement on incorporating this idea that there's places on the landscape that are likely to be more resilient to climate change into conservation and management decision making. If we can identify where those places are, what would you do differently in that place compared to one that you knew was going to experience a lot of mortality the next time there was a drought? And can you incorporate that into a more holistic conservation strategy for the chaparral? Okay, so 3 take-home messages, and the first is that we have models of persistence and resilience in the face of environmental change. Here, of particular importance in Southern California are places that experience less drought stress or drought refugia. The islands are an example. There are other examples on the mainland as well. And that plant functional types, especially hydraulic traits, can allow us to directly measure vulnerability and how it varies across the landscape. And this is an incredibly important tool for continuing to understand vulnerability in the face of change. And then finally, that drought refugia provide opportunities for us to build persistence and resilience into regional management strategies. So that's all I have. Thanks.
Has a couple of minutes for questions if there are.
How much more role do you think marine layer plays in that drought resistance? You know, that is much more persistent on the island than it is. I mean, we have it very persistent in some coastal areas of California, but not as widespread as we're all.
Yeah, so on the islands in particular, the maritime influence is, I think, the dominant factor there. There's other factors that could be differences in land use on the islands that could be contributing to that buffering effect as well. But as this figure shows, there are other places, riparian areas. There's some good research showing that demographic patterns of oaks tend towards these more mesic riparian microsites in southern parts of the range of particular oak species. So other places besides the islands are also likely to function in a similar way. And it's all has to do with availability of water and the ability to maintain favorable tissue water status during drought.
Time for one more.
So if you have a range of a particular species, parts of that range are still going to be, quote, more harsher compared to if that range extends to the coastal fog belt. But then you're going to lose the distribution in the areas where it's hotter and more extreme. How does that, but then eventually you'll get, I would think, some speciation. So I'm not sure how that helps. That refugia helps the entire range, or is it just a description of the narrow part of the range?
Those are great questions. I don't have good answers for them, but I do think that one of the realities of climate change is that we are going to be creating more disjunct distributions within species ranges as things retract into these refugial habitats. How do you maintain connection between those? How do you maintain gene flow between those? Those are really good questions for the future.
Thank you.
Thanks.